52 people, 53, yay, who listen to us. And it is 10:01 here on the Pacific Coast in California. We are at UC Davis, and I can already say welcome everyone who is joining. Welcome to our monthly West Coast Metabolomics Center seminar, where I am very pleased to welcome today Dr. Catalina Cuparencu. She is an assistant professor in the Department of Nutrition, Exercise, and Sports at the University of Copenhagen. And she loves that city a lot because she also did a postdoc there at University of Copenhagen with Professor Lars Dragstedt. Lars Dragstedt is very well known internationally in the area of food biomarkers. She also did before, from 2015 to 2019, a doctorate with Lars Dragstedt. And from 2011 to 2013, a master's. And before her master's, she had a bachelor at the Faculty of Pharmacy, University of Medicine and Pharmacy in Bucharest, Romania. So there's one time she changed her, her location. Her research combines experimental nutrition from human intervention studies with state-of-the-art metabolomics to improve the accuracy of dietary assessments through development of food biomarkers or biomarkers of food intake, but also to investigate the effects of dietary changes on metabolic responses and diet microbiome interactions. For example, for the prevention of cardiometabolic diseases. So she is professor since 2021, so it's fairly newish, but she has already, 3 years, right? But she has a number of good, really excellent papers. For example, a Nature Metabolism paper this year, Towards Nutrition with Precision, Unlocking Biomarkers as Dietary Assessment Tools, that has already been cited, but also a comprehensive database for DNA adaptomics, or the anserine to carnosine ratio, an excellent discriminator between white and red meats, or an untargeted urine metabolomics approach for autologous blood transfusion detection, also for differently cooked potatoes, for seafood intake compared to meat, for more red meat intake, and so on, and also for pea. So there's a lot of things that she has done in the past, and we are looking very much forward to the talk today by Catalina Cuparencu. Catalina, the floor is yours.
Thank you so much for a nice and thorough introduction and for having me. I'm actually assistant professor from this year. I've been postdoc until January this year. So I'm very fresh assistant professor, but nevertheless, I intend to go up the ladder as soon as possible, or, but it is my pleasure to present to you some of the work we've been doing here at the University of Copenhagen. And as you said, I've been working with biomarker development ever since my master project, when I first heard of metabolomics, and I realized the many opportunities metabolomics can bring to nutrition research. And biomarkers of food intake are one such opportunity, and my interest lies in their implementation in nutrition research as compliance measurement in dietary intervention, but I'm also overall interested to solve some of the most challenging parts of BFI development. So in my talk today, can you see the pointer?
Yes, we can see it.
Yes.
Thanks. Perfect. So in my talk today, I will start about talking about introducing dietary assessment and why this is a problem and how we can deal with it. And then I will skip into biomarker validation more than discovery. I will also go through the key concepts for biomarker implementation that we put out in our last Nature review, as well as describe the roadmap and how we can actually move forward from here. So in dietary assessment, I will start with the state of the art, and then I will introduce a bit the biomarkers. And of course, when we study nutrition and we do nutrition research, the one key, most important question is, what do people eat? Measuring what people eat is central. And it's not really easy. It's notoriously challenging. So even if you would try to recall, for example, what you had for lunch on Monday, and you would take a moment, is it easy to remember? How much details do you remember? Do you remember only the key foods? Do you remember at all? So usually in nutrition, the dietary data is self-reported data, the data that comes from the participants themselves, and recall is incredibly difficult. There's also, beside this, there are social norms, so people know what is healthy, what is not. They participate in dietary interventions, and when they have to report their food intake, they sometimes tend to overestimate what is believed to be healthy and underestimate what is believed to be not. It's also an extremely tedious process. So when you have to participate in these trials and even apply the state-of-the-art techniques, which means weighing the food you eat on a scale and registering it, they also tend to simplify their diet to, of course, make it easier on themselves. So currently we use food frequency questionnaires, which by their name measure the frequency of food consumptions on different foods and food groups. We work with, and usually this measures throughout half a year or a year. So on a longer period of time, we get a bit more information about habitual diets. But we also work with recent intake. So we have 24-hour recalls where participants have to record the diets that they had yesterday, or dietary records, which are prospective. So you start in the morning registering everything you eat. In most studies, working with so many different intervention trials, even when you advise participants to actually register through dietary records, so prospective, they still recall what they did yesterday, or they do it before going to bed. So the gold standard for dietary assessment are these 7-day weighted dietary records, but commonly accepted in nutrition research are also 4-day dietary records, where we usually work with average. So we want to have non-consecutive days that are recorded and some weekdays and some weekends. This is what we have. Now, a lot of the focus is put on the volunteers, how they misreport either intentionally or unintentional. But there are also many more other sources of error when translating dietary intake. So you have this record, then you have to code it, be it the dietitian or the volunteer. You have a software, you eat, a yogurt from a specific brand with a specific percentage of fat, and then you have this drop-down menu with many different similar yogurts. And then there are some brand names, but you don't find yours, so you try to then record the yogurt that seems similar to the one you had. So it's many different types of decisions. Now, the new softwares behind have a database in certain countries that are more updated, but other databases are also rely on really old food composition tables. Once the data is typed in the software, then essentially we have an Excel with dietary data that is the output. And there is some sort of software manager that at some point decided how to actually categorize the different foods into nutrients. But when you try to look more at the food groups, it's a bit more complicated, particularly when you have to deal with the different food like composite meals. So you have complex meals, and then you have to separate those into different food groups. This usually works better at the nutrient level, but at the food level, it becomes a bit more complicated. The new advancements in dietary assessment, wearables, camera-based, sensor-based, they are better. They are decreasing the tediousness of the process, but essentially they rely on the same softwares and on the same databases. So that still makes them a bit inaccurate, or depending on the research question, can still have systematic and random errors. Now, we like metabolomics, we like biomarkers, and we like them because we can measure. We consume foods, we metabolize it, and then after metabolism, we excrete in excreta such as urine or blood metabolites that are related to the food we ate. So by investigating these metabolites, we can trace back to figure out what food was consumed. And a simple search on PubMed with biomarkers of food intake will give almost 9,000 publications in the past 15 years. So biomarkers of food intake have received considerable attention, mainly probably because of the expected impact. They are expected to have a profound impact in nutrition research. And we can see even more and more interest from the more traditional nutrition professionals. So how can a biomarker-based dietary assessment help nutrition research? First of all, it will improve the reliability, which in turn will increase the trust in associations between diet and health. So when we have these diet-health associations with hazard ratios of 1.1, 1.2, we really cannot exclude other confounders. So it is overall really hard to draw causality and in turn inform dietary recommendation. A more precise dietary assessment will also enable more precision in these dietary recommendations. Biomarkers also bring the hope to supporting this paradigm shift in nutrition, moving from nutrient status into measuring more intake of individual foods. And this is important overall because it's simply easier to base dietary recommendations targeting specific foods rather than level of nutrients. Of course, micro and macronutrients are important. It's more how do we actually communicate the importance of dietary guidelines. Now, just to take a step back, what are biomarkers? And biomarkers are objective measures used to characterize the current condition of a biological system. So we have a biological system, let's say the human body, and we have different types of exposures. We have physiological, chemical, social, as well as environmental, food, and diet. And so this exposure will have an effect on the biological system, and depending on the susceptibility of the biological system or its resilience to change, this may modify the strength of this effect. So basically, different type of exposure put a pressure or a challenge on the health status, and it is the resilience of the biological system that defines the magnitude of the effect or that the exposure had. Of course, the human body is also a dynamic system, so it also feedbacks to the change, and overall the susceptibility changes over time. Well, based on this and with this in mind, in the football project, we developed a flexible classification of biomarkers according to these three types. So we have biomarkers of exposure or intake, we have effect markers, and we have susceptibility biomarkers. So the exposure markers relate more to the current intake. There are nutrient intake biomarkers, non-nutrient food or food components, as well as biomarkers of dietary patterns. When we talk susceptibility markers, these are essentially reflecting the current status. So they are nutrient status biomarkers or non-nutrient status, host factor biomarkers, and so forth. And then we have the effect markers that are essentially reflecting a change. That's the effect. So what is important from a biomarker's perspective is that the same molecule can act as any of these three markers depending on the context. So if we are to take, for example, glucose, the same glucose assay, right? If you eat a slice of cake, glucose rises immediately after intake is an exposure marker. If next morning you take a fasting glucose, and you measure the levels with the same assay and the glucose levels are still high, that means that your body cannot dispose of the extra glucose. So that's a susceptibility biomarker. But if you change your diet and you want to measure how that diet change affected glucose, you do it again in a fasting morning sample, and that would be an effect marker, and that is the effect of the diet. So essentially, it's the same assay. It's not the biomarker per se. It's the context. And this work has been spearheaded by a former PhD student in our group, Qian Gao, together with our professor, Lars Dragsted. But really, the contribution came from all the PIs in the football project. And these are people that have been working with development biomarkers of food intake ever since from the beginning. Now, yes, we do biomarkers, but of course, it's not so easy, right? So measuring diet, it's certainly not an easy task. Diet is not one food, it's many different food groups and subfood groups, and essentially we don't even consume food groups or foods, we eat complex meals, so we eat the combination of this. So if we are to zoom into one of these food groups, whether it's the food of animal origins, then here we'll need biomarkers to differentiate dairy products, egg and meat. Within there, we have milk, butter, cheese, yogurt, so we have fermented, non-fermented. Some of the eggs are enriched with some other micronutrients. And within the meat, it's even more complicated because, we are meat, we burn our own muscle mass and we eat muscle meat. So when we try to find biomarkers of meat, it's really important to distinguish for those that are that arise endogenously. And then we have to also differentiate between the different sources of meat, of which some are believed to have different health effects than others. We're also interested in different processed meats and heated meats. So there's a lot to look into only in one food group. So developing biomarkers for all these foods requires really many studies. Now, there is a really great talk on the same topic that was part of the seminar series around 3 years ago. And Professor Lorraine Brennan from UCD in Dublin talks very much in detail about the biomarker discovery strategies. And she also introduces a lot of the dose response studies, which have been a lot conducted in her research group. So I really encourage you, if you're interested in the topic, to also give a listen to this talk. But just really briefly, to discover biomarkers, we conduct meal studies with 1-2 foods at a time, and we collect urine and blood samples up to 48 hours. Then we have dose response studies that we can conduct with few foods at a time if we logistically organize well, and then we try to then predict intake either with or without amount consumed in cross-sectional studies with habitual diets. Now, moving into biomarker validation, and through the biomarker validation, I'll also talk a bit about discovery here and there. But so biomarkers as a dietary assessment tools are not effect markers, and they reflect exposure to foods, and ideally they are non-nutrients. Ideally, to simplify how complicated things can get. And ideally, they're not useful for the human body, so the least interesting and the least metabolized and the easier to measure. Now, also within football, we created a framework to actually describe what is a good biomarker, because there are so many, in so many papers, are they all equally good? So we developed a framework to critically validate biomarkers of food intake, and this framework contains several biological aspects and some analytical ones. So we're interested in plausibility, its dose response, time response, robustness and reliability, as well as how stable it is, how does it perform analytically, and is it reproducible? So I will walk you through one by one now. So plausibility means that the biomarker is chemically related and specific to the food of interest. So we discover biomarkers either in randomized controlled trials, meal studies, most of them, but some discovery also happens from observational studies. It is really important to interpret these biomarkers, essentially to prune the list of candidate biomarkers, and that also allows us to have a thorough search of potential confounding sources. The challenge with this criteria is that many biomarkers make sense, they are chemically related, but most of them are not specific to one food. The next criteria is dose response. So here we want the biomarker to increase with increasing intakes. And so the way we validate this is we run dose response studies. Based on that, we calculate calibration curves, and then calibration equations, and then with those equations, we predict intake in independent studies. And this is an example for proline betaine, which is probably the best biomarker of food intake it exists. It's a marker of citrus intake. And then you can see a really nice linearity between intake and the concentration of the biomarker. And we have there is a handful of biomarkers having those responses, most don't, or is not studied. Now, the next criteria is time response, and this is something that we figure out, we characterize in... Sorry? This is something that we usually characterize in PK studies, so in pharmacokinetic studies, in this postprandial randomized controlled trial that we conduct. So the time response is really important to determine the half-life of the metabolite, its excretion kinetics, and whether it saturates with repeated intakes. You know, if you consume several times the same food, will it then eliminate and go up again, or will it just build gradually? Now, here are two examples. And we have on the top carnosine. So both of these are marker of meat intake, and this is data from a study that I will come back later. But what we see here on top, so the meal study looked into 3 different types of meats, chicken, pork, and beef, versus a control. So what we see here from a kinetic point of view is that the carnosine goes back to baseline levels already of 12 hours, whereas this other one, 3-methylhistidine, which is usually a marker of chicken intake, is still elevated, even way beyond 12 to 24 hours. That means that if on a random day you measure 3-methylhistidine in urine samples, you are uncertain. It could be that intake comes from the 2 days ago. But if you measure carnosine, you're certain that meat was consumed in that day because this metabolite is out already at 12 hours. The next criteria, robustness. So here we want to show that the biomarker works as part of a varied diet, mainly because we discovered these biomarkers in these very highly controlled settings, and we want to use them actually in real life. So as a minimum, we want to reconfirm these biomarkers in similar studies, but ideally we want to measure them in studies with contrasting design. So we want to move from RCT to observational and vice versa. The last one, reliability, refers to that the biomarker has to be comparable with other dietary assessment, other dietary instruments. And as a minimum here, we want to associate biomarkers with reported intakes. But again, ideally, what we want is to predict intake. So to use samples from one study and predict samples from another study. Now, the accurate reliability, so how to best test this reliability, relies on the comparator. Because when you compare a biomarker that you don't really know how it performs with a dietary registration that you don't know how accurate it is, when something doesn't match, you don't know whose fault it is, right? So you have to do this many different times, or at least a few times. And it's also really important, again, the excretion kinetics are important because if you measure it in the wrong sample, then you will not measure it, not because it was not consumed, but because it was the wrong sample. So for most biomarkers require dietary registration to be concurrent to the biological sampling because most reflect very recent intake. But we're going to get to this a bit later as well. So I'm going to show you now a bit of a strategy for testing some of these criteria, and I will take as an example red meat biomarkers because this is what I actually did in my PhD. So here we worked with the strategy to discover, confirm, predict, and predict again. So we started highly controlled in a 4-way crossover randomized trial, and then we loosened up a bit. This was a not controlled studies with just a lunch intervention, and then essentially we used this optimized biomarkers to predict intake in a fully free-living population that had no control at all. So we moved from randomized control trial to cross-section. So the discovery study was conducted in 10 healthy volunteers. Usually these discovery studies are in a small sample size because we have the crossover advantage to it. And so here we had the restricted diet. For 2 days, these people were not allowed to consume any meat or fish, not processed meats or nothing. And they also had a standardized dinner just before the test day. And then they came in, and depending on which arm of the intervention they ate either meatballs with beef or pork or chicken, or some meatballs that were made from green pea with egg white. So it had all the macronutrients aligned, but that's also not really important for the biomarkers that we're interested in. And then we collected urine and blood samples up to 48 hours, and they were also supposed not to eat any of the similar foods up until collection was over. So the point here, the aim was to explore biomarkers related to the heme iron content, but we also wanted to investigate different meats simultaneously because we figured that that was a big gap in the literature where you wouldn't really have all of them together. And sometimes you also don't know about pork. Is it, more white meat or more red meat because it's kind of more pink? Then we apply the usual untargeted metabolomics pipeline. We run here a Qtop-MS. We pre-process data with MZ-mine, and we ended up with 6,000 metabolites in positive and negative mode. Then we had different data analysis strategies. So PLSDA comparing the meats with the controls, PLS to predict this heme, the vector as a heme iron content that we measured in the meats. And then we had to weigh ANOVA to look at the interaction between time and meals. And we only selected metabolites that were picked up by at least 2 of these methods. We ended up with 21 putative biomarkers, of which 9 seemed characteristic of beef and pork, and there were also some that were characteristic of all 3 meats. Out of this, we identified 17. And the beauty of metabolomics is that you can measure so many different compounds originating from so many different sources. So we have some biomarkers, a range of dipeptides coming from collagen. We have some meat flavor and other types of flavors. We have amino acid metabolism and muscle breakdown, thermal degradation, and even animal feeds, surprisingly. So these animal feeds metabolites are essentially isoflavones and are soy characteristics. So our interpretation was that they were coming from soy meal animal feeds, and then we discarded because, of course, they are not, plausible to be markers of meat. Now, looking at the kinetic profiles, we see, the collagen-related, unsurprisingly, has a high baseline, despite these people not consuming meats for 2 days in advance, because we burn collagen ourselves. This is a very similar story to carnitines, for example. Then we had the jackpot marker of red meat, this thiophene mercapturate, and we were very happy about it. And then we have this carnosine and anserine, really well-known meat markers, already existed in the literature. But what we could observe here was that the different meats have actually a different response. So for carnosine, pork is a lot higher, and beef is very similar to chicken. Whereas for anserine, a well-known chicken marker, you can also see that it's elevated from beef and pork. So now I have 14 candidate biomarkers that are plausible and have a kinetic profile that makes sense. Question is, can we use them forward? Are they robust and reliable, and can they actually predict meat intake? So with this, we went on into another study, which was this lunch intervention. Only 6 participants, but good enough. So these people had no restriction before the test day, except for no meat and fish during the sample collection. And then we had, it was not randomized. So the first lunch, it was a vegetarian lunch. And then the second day, they had a beef steak with French fries and some sauce. So then we applied the same untargeted metabolomics pipeline. And then we were really inclusive in the pre-processing to make sure we get as many of them. And then we tried to match these 14 plausible metabolites, of which we could only replicate 8. Some of these were not retrieved in the dataset, and some metabolites, unfortunately, did not show consistent kinetics. And this is where our jackpot red meat marker went down, because you could actually see that without a restriction, this marker was already elevated. So there are certainly different other sources. Some other biomarkers, this is carnosine, replicated very well. So we continued with them. And then we tried to predict meat intake in this study. So for this, we use PLSDA. And how this works is that we calibrate the model based on the samples from one study, in this case, the 4-way randomized controlled trial, and then we predict the samples from an independent study. And the model, we feed the model with all this. So for this model, we took all these 6 markers that are reflective of red meat and all meat. We didn't take the white meat markers because we thought it didn't make sense. And so what we want here, it's basically you calibrate, and then it's a pattern of metabolites. So for the meat, for a meat sample, you would have a higher concentration of carnosine, and for a vegetarian sample, we will not really have carnosine, right? So if you get in the model a sample that has high carnosine, then you want it to predict is it meat, and if you get one without, then it should be a salad. If you predict the other way around, then you have a misclassification. So we then evaluate the model performances based on the lowest misclassification rate of the test set, and then also the area under the receiving operating curves of both studies. And then we're also interested in taking a parsimonious model, so taking a model that performs as well, but with the least number of metabolites. Yes, so we did this prediction in the lunch meal study, and then we actually saw that carnosine was very good at differentiating. So you have here on the right hand side the predicted values. So these are from the second study, and these are from the highly controlled one. So carnosine by itself was discriminating well, the second study, but not really the first one. I mean, here you have an area under the curve of 0.7. However, when we combine carnosine with another metabolite, then it was a really nice, clear separation. So what we could see here was that in order to classify red meat intake, combined markers were more reliable than single markers in this case. And then we tried all possible combinations of these 6 markers, and then we could see that one marker was never selected by any prediction model. This proline, hydroxyproline, which is the carnitine like the one with higher baseline, that was selected by 2. We have another one that was selected only once, but the same model was as good without it, so it's not a good predictor. So essentially the best markers were creatine, carnosine, and this mehialanine. Also, the sad fact is that was for my PhD that none of the reliable markers were actually red meat markers. So in order to fully test reliability and robustness, then we worked with, we looked into this previous study, and that is, the previous study was a 3-year, 7 multi-center lifestyle intervention where the participants were supposed to eat either a moderate protein diet or a high protein diet with different combinations of glycemic index and physical activity, basically to try to find the best diet to maintain weight loss. But within this study, we could run a cross-sectional sub-study, and then we found 130 participants from a center in Zealand that actually had matching dietary registration with 24-hour urine collection. And then in that software, we could also go in detail to not only look at the average between the 4-day dietary records that usually the study was working with, but to look more at the day level and the different foods that they consumed. So it was a really nice data set to actually play with. And just to illustrate a bit what the habits of meat consumption in this subset was, so we had this 130 participants. Some of them were seemingly vegetarian, so it doesn't mean that they were actually never eating meat, but in those days matching the biological samples, they didn't eat any meat. There were some that only ate red meat, some that only ate poultry, and the majority that ate more than one meat. And you can also see this very nicely distributed on the PCA plot here. So, you know, the straight line is only fish or poultry or red meat consumers, and what's in between, they consume 2 of them at least. So we did, again, the same type of PLSDA, again, combination NK, so trying to test all the different combinations between these markers. And here we added the anserine and methylhistidine as well. So we kept those 4 markers that were better predictive of red meat, and we added them with the white meat markers. And then what we could see actually was that anserine alone was very good at predicting the vegetarians. So if you never ate meat, anserine was very good at predicting that. It was pretty good also at predicting total meat eaters. So here we combined consumers of red meat, poultry, processed meats, and fish, everything together. And we had 17 samples that were misclassified. I mean, not too bad. But then, of course, my interest was still red meat markers. So the next step here was to try to look into this population that I knew actually consumed meat and trying to to work with the different with the discriminant analysis between chicken and pork or chicken and red meat and see if I can actually do anything about that. So basically here again I fed all the different the model all the different markers, but then here now we didn't compare meat with vegetarian but meat with chicken in both studies. And what we could see was that the ratio between anserine and carnosine was actually a very good discriminant of red and white meats. And so we didn't have so many red meat consumers, so only 18, but the white meats again were well classified. And interestingly, 4 red samples were always misclassified by all the models. So we don't really know whether those were miscompliant or whether the markers are not performing as well for the red meat markers. So this is essentially to tell that from the candidate biomarkers, and these are even the 14 optimized ones, only 2 are actually predictive of intake in cross-sectional studies. So validating biomarkers for all the different robustness and reliability, it's really, really essential in actually pruning the list down to something that we can then measure as part of, the big panel for all the diet. There are also some validation criteria covering analytical aspects. So we have stability of the markers. So these biomarkers must allow repeated measurements with no sign of degradation. Usually we have certain samples collected in cohorts that we freeze 10,000 times to do different analysis. So it's important that they actually stay stable to overusage. The analytical performance usually for untargeted metabolomics is fine, but for targeted one, we need LOD, log, and internal standards, and as well to test the accuracy and specificity and precision of the methods. And then we want them to be reproducible between labs. So ideally, it's not only one lab that has the key to everything, but we can actually have standardized methods and across labs. Now, so all these criteria are came from the football project and are under Dragsted's paper from 2018. Now, in the review that we just put out, we also included an additional validation criteria, and that is the biological variability. And we felt initially that some of this was already covered in robustness, but essentially one of the reviewers pointed out very well that this is actually largely unexplored. So we were very happy to get those comments and end up including this to, for the benefit of everyone. So the biological variability means that the biomarker should have a low individual variability and to the same amount of food consumed. And here the trick is that we really don't know. This is largely unexplored, particularly in experimental studies, because usually we have one meal study where the participant eats one meal at a time, or we have a bit of a dose response, but we don't really redo the same study. So we don't have participants eating the same food, the same amounts several times. So it's a really virgin land that has to be explored a bit better. Some of these methods that can measure biological variability are the ICCs, and they are used to assess this variability longitudinally in observational studies at the moment, but they're also influenced by half-life, frequency of intake, and overall the dietary habits in the population study. So we don't really know if this is the best measure. So far, it's the only one used. There are additional tools coming out, like metabovariant 2.0, also from Lorraine Brennan's group. But essentially, experimental studies looking into this are lacking. So the implication of the validation framework, should all biomarkers fulfill all these criteria? Ideally, but what's important to note is that these criteria are not meant as a score. So are mainly essentially meant to characterize the biomarker. If we know that the biomarker is very robust and reliable in predicting intake, but it doesn't have a dose response, maybe we can use it in a study where we only need to assess compliance. So we need to better understand the biomarkers we're working with to find solutions for their implementation. Elimination kinetics are central to the application of biomarkers. And this is essentially because it determines in which type of studies to test the robustness and reliability, and also it tells in which samples you actually measure them. So from this perspective, biomarkers that are only discovered in observational studies but not confirmed in randomized controlled trials are not truly informative. So they need a bit of extra work. And certainly different studies are needed to fully validate biomarkers according to all the criteria. So we need discovery studies, confirmation, and predictions. Different and similar and contrasting designs. So I will move now to some key concepts for biomarker implementation. And actually, I will start with the status of biomarkers. I forgot about that. But so in football, we basically put out the most important food groups. And based on that, and based on these biological criteria, then we did a systematic research of the literature. And then we ended up with really very many, really great, but also really long reviews. I think everyone in the project was a bit overwhelmed at the end of all this journey. It's a lot of structured information, but still very difficult to navigate, and to really, even for us, it was hard to get a sense of what we actually have, what can we use, what do we do. So the main challenges were that most biomarkers reflect only recent intake. So how can we even use them to measure what people eat habitually? Most of biomarkers are not specific, and there are very few quantitative biomarkers. So after a good break, we sat in our research group, and we tried to brainstorm a bit how we can we actually accelerate biomarker implementation. So for this, we just developed a new score, a new framework. But this is still based on the same criteria. It's just that it's tried to put a bit more emphasis on which criteria are really useful to start implementation. So we use the plausibility, robustness, and reliability as the most important. And if these are fulfilled, then a biomarker is specific to the food of interest. It works as part of a complex dietary matrix, and it's also comparable with other dietary instruments. Ideally, it's also good at predicting intake. If it's level 2, it only misses the reliability part. And if it's only plausible, then there's quite a lot of work. There are also some biomarkers that, they're not plausible, but they're actually very good at improving the prediction in combined markers. So we also try to rank those. And so based on this new scale, we did another systematic search, but this time we actually had all the BFI reviews that were conducted up until 2018, 2019. Then we looked at all the intervention studies, because also in football we ran quite some meal studies. And then we looked also at observational studies from earlier on. So based on this, we realized that actually, with a little effort, validated biomarkers in urine will reach most important foods in the human diet very fast. So we already had utility level 1 markers for meat and fish, chicken, some processed meats, fatty fish. We have some fruits, the citrus fruits, banana. We have whole grain, wheat and rye, alcohol, and some beverages. But also plant foods, vegetables, some other fruits, legumes and tubers and dairy are not really far. So these compounds, these metabolites only require to be tested for reliability, which essentially means that if we have 2-3 good meal studies and some nice cross-sectional studies matching dietary records with the biological sampling, you can do this reliability pretty fast. So it's not that bad. Now, moving forward to additional concepts for biomarker implementation, the first one concept we are really keen into putting out there is that the biomarker elimination kinetics determine when to sample, the time window of sampling. So we introduced this concept, which is, we think, a bit easier to understand than half-life. So it's the same idea as we looked what I presented earlier. So we consume the food, it's metabolized, and we have quite some factors affecting individual variability. Probably ADME is really important at this stage. And then we have different types of compounds. So we have carnosine that is out at 12 hours, but we also have markers that we can measure up until 48 hours. So this is hypafferine or tryptophan betaine, and this is a marker of lentils, also reported for some nuts. So what's important about this is also biomarker kinetics in relation to the frequency of food intake. So here we're going to start with once per week, and then here we have 5 different metabolites with a half-life of between 1 and 24 hours. So essentially, the half-lives of 1 to 6 hours, these can be measured up to 1 day, as you can definitely see here. Actually, the first one, you can't even measure it in 1 day, but for the 12 hours, you can measure it up to 2.5 days. And for even longer half-life, you can measure up to 5 days. So I think this calculation is based that you can measure up to 5 half-lives in blood. Now, this is if you eat once per week, right? If you eat the same food 3 times a week, then it's easier to measure it. And if you eat something daily, there's no problem. So the main problem is to those foods that have a very short half-life and are not consumed very frequently. So the implications of biomarkers time windows of sampling is that to validate reliability, we need to have matching schemes, and most of the current biomarkers have this time windows of sampling between 1 and 3 days. Actually, in the paper, in the Nature review, in the supplementary material, we also put in a time window of sampling for all the metabolites that we would actually find estimated such samplings windows. It's also important to interpret with caution when biomarkers are measured in random samples that are collected in observational studies that are not matched to dietary registration, because it's really by chance. We don't really know what we're measuring. And also importantly, because in many dietary intervention, we have before and after samples, and these are really good at measuring the effect of whatever intervention it was, but they're not sufficiently good to measure biomarkers. These biomarkers would essentially also only reflect the time window that they reflect. The next concept is the multiple samples to capture long-term dietary intake. So what we show here is that with the same type of window of sampling, if you have a marker with a 24-hour window of sampling, you have a likelihood of lower than 40% to detect these biomarkers in 1 urine spot. But even with this marker, if you collect three samples, then the likelihood already rises to 80%. So essentially, if we are to develop more effective sampling strategies, so remote spot urines that people can collect at home, we can also more easily implement a multiple sampling to actually cover the diet in the long term. Last but not least is how we select and combine biomarkers based on the intended dietary assessment. And here, just to take one step back where we started, we have food frequency questionnaires. These are indeed looking into habitual diet, but the other most, gold standard are still reflecting recent intake. So what we do with this is that we collect more of them, and then we take averages, we calculate averages, and we make assumptions. So we assume that those for the dietary registration are valid to represent the whole year, or sometimes we have baseline dietary records, and we associate it with lifelong diseases, and we assume that the diet was the same throughout. So we're a bit unfair to biomarkers asking that they should tell us everything, one biomarker per food. So in that sense, it's as we do with the tools that we already have, we should also try to implement biomarkers based on their intended use. So essentially, we have 2 main questions. In nutrition, we want to evaluate habitual diets in epidemiology, and we want to assess compliance in dietary intervention. For the first one, for habitual diet, We're interested indeed to assess long-term intake of a specific dietary pattern. And so for this, we want to have validated biomarkers of key foods and food groups, ideally that are quantitative, at least to indicate high and low intakes. So these biomarkers should indicate habitual intake, and preferably biomarkers should have longer half-lives in studies where only one or a few samples are collected per participant. However, for compliance in dietary interventions, we again need validated biomarkers and sufficient samples, but qualitative biomarkers are fine. If you have an intervention where you just want to test if the participant actually ate the food you gave or didn't eat the food that you said they shouldn't, qualitative markers are good enough. So as long as they match the sampling scheme and we know what we measure, that should be fine. So here is work. I present some work from Natalia Vasquez Majares. She was also a PhD when I was a PhD. Now she's in Mexico running her metabolomics lab. But here her work was on banana biomarkers. And so here she also looked at different combinations. So we have similar approach between the meat and the banana. And what she actually did was try to classify with PLSDA non-consumers from low consumers and from high consumers. So in the same approach, we have a highly controlled study here and the cross-sectional study, the Carmen study from Germany here. So what we see is that the high consumers of banana had the lowest misclassification rate, only 7%, but it was harder to, but the no consumers had a higher rate. And when looking into the low consumers, these were actually harder to classify. But nevertheless, and probably this is a limitation of PLSDA in general, because it can only look at two classes simultaneously. So looking at this with a different analysis could also point towards a different type of dose response. For the meat, we looked into a different approach. So we made a stepwise assessment, was any meat consumed at all? If it was, then what type was it? And for this, we used answering to carnosine ratio, but in similarity, we didn't have the data for it. But in similarity, TMAO and carnosine should also work to differentiate between meat and fish. And when we actually knew which participants ate red meat, then we could also use carnosine to indicate how much of it. Again, low consumer, no consumer, high consumer. So based on these different ways of combining markers, we also suggest how to combine biomarkers to measure different intakes. So if we want to measure intake of a single food or food groups, and again, I take meat as an example, we can measure anserine to measure chicken. We can measure anserine to carnosine ratio to differentiate red and white meats. And if we want to measure all meat and fish, then we can measure all the biomarkers. If we want to measure intake of a whole grain chicken sandwich with cheese, then we can measure biomarkers for whole grain, biomarkers for chicken, and biomarkers for cheese. Whereas for dietary patterns, we can measure the markers for the key foods in the diet. And here the dietary patterns can vary. So you can use the same approach for the different diets you're interested in. Now, biomarkers also allow to something more, actually, to also include metabolites that are reflective of production of our cooking processes. So I think this is probably the first study we published from our group, from GIOSDE, that used this combination of markers and trying to find the parsimonious model. And so in the unique pattern of metabolites reflecting intake of beer, she had markers that were reflecting hops, so raw components from beer, such as iso-alpha acids and tricyclohumols, and also brewing components. In similarity, we also looked, so this is the same study as the meat one, but it was a side dish. So sides to the meats, we had different types of potatoes. We had rice, French fries, boiled potatoes, French fries, and chips. And so here we wanted to look at different cooking or deep frying markers and overall markers of potatoes. And here we nicely saw furans and pyrrolines and pyrazines, which are highly heated metabolites of highly heated processes, so Maillard reaction. So you can see here the difference between French fries and chips is that the French fries are the finger potatoes and the chips are the flat ones, like the Pringles, just because different cultures have different interpretations. So you can easily see the nice dose response increase in this deep frying metabolites, which also makes sense because the French fries are, the surface of being deep fried is smaller than the flat ones that are fried everywhere, basically. So this can then be added further in the combined markers models. And this is some level of detail that we rarely actually get from the current dietary assessment tools. So take-home messages. Biomarkers of food intake should be clearly differentiated from effect biomarkers. Most food groups will soon be covered in urine by reflecting recent intake, was it consumed or not, and maybe high and low intakes. And these qualitative markers can solve many of the existing questions that we have. To be able to use them in proof of concept studies, we need to have sampling schemes that match biomarker time windows of sampling. And ideally, we start collecting multiple samples throughout the year in order to cover long-term dietary intakes. Now, lastly, roadmap moving forward. So here we try to put together a roadmap for biomarker development. So looking not only into discovery and validation, but also into the next steps that are already needed to prove that the concept actually works and is valuable for nutrition research. We don't really know. So proof of concept studies are equally important at this point. So I will briefly walk you through this roadmap. For further biomarker development, discovery, and validation, we are interested in single and combined markers for food groups, and dietary patterns. But here it's also important to speed the process up a bit. So when we conduct these meal studies, it's nice to already have some cross-sectional studies where we can try to evaluate prediction studies. Then we optimize the biomarkers faster. It's also important to develop biomarkers to cover the missing foods in the diet. But in order to more efficiently do this, maybe a focus should also be on contrasting between foods. So newer dietary recommendations are telling us eat a bit more of this and a bit less of that. So already when we select the control foods in the meal studies we conduct, maybe having this into account, so looking at refined versus whole grains, or similar contrasts would be ideal. Then, of course, we need to cover dose response and biological variability. And we probably need to think of some good cost-effective ways. And we need more biomarkers in blood samples. We hope maybe that in blood samples, we can also find biomarkers with a bit of a longer half-life. That would also help. Now, for biomarker implementation, we need proof of concept studies. And there are not really so many studies out there, but there is this one study that was recently out, and this is from the group at the University of Barcelona. And so here they actually show the study is about linking dietary fibers with cardiometabolic risk factors, and they have an approach where they use metabolomics-based predicted fibers, so based on biomarkers, with self-reported. So here you can see that there is a significant risk of reducing C-reactive protein compared to the self-reported, which is also things overall you can see that the results you get from biomarker-based prediction are quite different from self-reported. We never know if it's going to be good or bad, but it's important to actually get some of these studies out there and try to figure out in which direction we're going. So I was very happy to see this study. So here the list.
Come to an end, please, Catalina.
Yes.
We come to an end?
Yes. So here the list would be to introduce biomarkers in nutrition study to evaluate compliance, to develop algorithms to evaluate food frequencies and time since last meal, multi-marker methods for diet typing, biomarkers based, and we need to standardize these methods across labs. And not least, we need internal standards for true quantification. So not surprisingly, there is still quite some work to be done to fully implement biomarkers. But for this, I cannot stress enough that bridging efforts, working together, collaborating, cross-validating biomarkers will really be a great advantage. There is enough work for everyone. And not least, we need to be better at sharing data. So in Copenhagen, we're currently working on a data sharing platform where we that allows the cross queries across studies, and we hope that this will speed up metabolite identification and understanding of specificity. So I personally have little doubts that in 10 or 15 years, biomarkers of food intake will be applied at a large scale in nutrition research, most probably still along with some of the traditional tools. But I do see a future where biomarkers have a high chance to replace 24-hour recalls, and with sufficient development in remote sampling, the easiness to collect such samples will probably be decisive in influencing this. So with this, I would like to acknowledge my group here at the University of Copenhagen, particularly Blas Dragsted, but also Julia and Chian and Natalia and Gyozde that were part of this whole football project, and not least collaborators from different universities as well. And with this, I'm open for questions.
Thank you so much, Catalina, for a lively, engaging talk. I really loved it. Arpana, you would like to lead?
Yes, great. Thank you, Catalina. That was such a great talk. I learned so much about what you're working on. So starting kind of like at the beginning of your talk, you showed challenges of measuring diet through biomarkers with regards to complex meals, but you had said that some people believe there are differences in the type of meat you eat. Do you think the data is not strong enough yet to talk about the risk of red meat versus white meat intake?
The thing is, what is red meat? So red meat, is it a processed red meat or is it fresh red meat? In the fresh red meat, actually, lamb, and so there are different results for pork, lamb, and beef. And then meta-analysis actually show that. So if we put all red meat together, maybe, maybe not. We don't really know. So we are actually not able with the current evidence to know which type of red meat is it all. Of course, the whole topic,
well, good thing I'm a
it's a bit,
vegetarian.
the whole topic is a bit against all the dietary recommendations at the moment, but we're scientists and we really want to know what the science tells, right?
So kind of like like related to that question, do you think the recommendations for nutrition and like nutrition research will ever catch up, or is it kind of always one lagging after the other? What do you think about
Catch
that?
up with what? What do you mean?
Like how people, there's always recommendations of what to eat, what not to eat. And then there's the reality of nutrition research of what, you presented and what's currently going on. What are your thoughts on that?
I mean, this is a very philosophical question that I like, mainly because, even if we are to think of, everyone knows Mediterranean diet is healthy, right? And we can, wherever we are in the world, make similar diets. We also have in Denmark made, the Nordic diets. But the question is, why don't people do not eat it? And that comes to comfort, that comes to price, that comes to habit. So, in our little bubbles here in our offices of science, we want to, find the best, but essentially it's really interdisciplinary. So I think it's actually about time to combine consumer behavior together with sociology of food in bigger projects where we also implement a bit more and try to understand consumers on the other hand. Because I mean, nutrition will not give a magic pill or a magic superfood that everyone eats and stays healthy. That's not going to happen, right? So it's also very individualistic and very personalized. So there are different ways to approach, but you will never reach everyone.
Yeah, that's such a good point. Moving on, you had spoken about the framework on what makes a good biomarker. I loved the systematic approach that you had shown. What is the sample type measured for carnosine and 3-MH? I can't forget.
So everything I presented was urine. Everything I presented was urine, and Sorry?
Mhm.
And
Oh, yeah, go
overall,
ahead.
overall, the urine biological sample overall covers more biomarkers than blood does. That's not because blood is not as good. Actually, there's a really nice paper from Mary Pladen comparing blood and urine after a really nice study, and it seems that they're actually very comparable. But there are not as many, remotely as many studies or results published in blood yet. This is why that's also a big sign, because usually, you know, in all the cross-sectional study, we mainly have blood.
Yes. Okay, great. You had talked about the meat study, where you showed that you measured 6,000 metabolites. That likely includes unknowns. But on your next slide on red meat, you showed identified compounds. Do you ignore unknowns or do you identify them afterwards?
That's a good question. We report them. I did try to include most of the unknowns as well in the prediction models, but they didn't come up well. So it was essentially from 21 to 14. So 21, there were 17 identified, right? And only 14 that then had a good kinetics profile. So I think they also failed the kinetic profile afterwards. But usually we keep unknowns in the loop.
Okay. On those kinetic profiles, the y-axis showed a relative LCMS response. Why did you not try to rerun samples for accurate quantification once you knew the BFI metabolites to look for and to quantify?
So easily said. So at the moment we're working on, you know, targeted methods. There is no internal standards for most food biomarkers. So for true quantification, you need labeled standards, and those don't exist. So the best we can actually do at the moment is semi-quantification and have some of the internal standards representing different parts or different types of metabolites, and then work with that. But yeah, so currently there's one such method in the whole world, and that's in Barcelona, where they have a semi-quantitative method. And I know there are different research groups trying to set this up, because of course this is the next thing everyone is interested in, targeting, and then not spend so much time in doing all the identification, but be a bit more efficient and, you know, moving towards proof of concept studies.
Great. There are several general food biomarker questions. I think just because, you know, the group of people attending are from different backgrounds, I'm just going to go ahead and ask those.
Go ahead.
How can the food biomarker strategy be used to objectively address questions in diet questionnaires that focus on long-term food intake, such as did you consume peaches over the past 12 months, and how frequently did you drink tomato or vegetable juices over the past 12 months? So just thinking about, like, you know, our everyday life, we eat foods, you know, on occasion or seasonally, pumpkin lattes and all these things. Like, how does those, like you were saying, behaviors result in the long food intake studies?
So this is it's also, as I had one on one of the slides, the same problem we also have with current dietary tools, with the traditional ones. I mean, yes, we do have the food frequency questionnaires in some populations where these are well validated, they work well or better, but it's still very hard to approximate, particularly for low intakes, but also in general, everything is a bit of an approximation, right? But then we also think that the dietary records, 4 times the average of those, are reflecting, and the data, at least at the nutrient level, you know, the data says that they are reflective of longer term intake. For what I see, the future is multiple samples collected at home. So if we are to think that a 24-hour or a spot urine sample reflects the recent intake of 24 hours, just as 24-hour recalls do, that's actually easier to pee in a little tube and have it in your freezer and do that twice a month and to do that, twice a month for a year, and then collect those in one sample. That would be a lot more representative than what we do for the dietary averages. Right? So if we, and probably the same with blood, but I guess urine it's even easier. And considering that most biomarkers, more foods are soon going to be covered in urine, and the blood is still, hopping behind. That's how I actually see the future, or the near future.
Yeah, yeah. Doable at home for sure with
Yeah.
urine.
I mean, I guess also with
yeah.
sport urine samples, with dried urine samples, and as well with dried blood samples, but those also have their caveats, and you actually have to know the amount of sample that was dried in order to calculate concentrations and so forth. So far, some studies from UK show that sport urines before dinners or first void in the morning actually are pretty comparable with 24-hour urine. So why not just do many of those and see what we get?
Yeah, yeah. That's a good point. John, you have your hand up.
Yeah, I just wanted to ask a quick follow-up question on that specific question. So I really like the breadth of what you've been doing. But this temporal versus concentration aspect and the kinetics, etc. I was just wondering your thoughts on whether it would be worthwhile when you have those serial sampling of people, would it also be valuable or not to have two repeated measures on the same day? Because if you know the clearance rates, we're always going to be dealing with a problem of intake intensity and proximity of intake, right? Because of the clearance. So a concentration of a given amount could either be because they ate a lot or they had it recently. So do you think it would be feasible, useful to have essentially two samples in a day on multiple days a month, so that you could actually get some idea of proximity and intensity of intake from that measurement? And that just, what's your opinion?
Do you mean, so when you say two samples in the day, would they be collected at the same time, or one in the morning, one in the evening?
One in the morning, one in the evening, that kind of thing.
That can only, give more information to better understand.
Yeah.
Another way how we look at it in this term of proximity is also if you have biomarkers with different windows of sampling, combining those can actually tell when the food was consumed.
And when you mean windows of sampling, you mean because they have different clearance kinetics, that the optimal sampling would be at a different time? Is that?
Can you repeat that, please?
Well, I don't quite understand your answer. So you said,
if you have, so by proximity, you mean when it was consumed, right?
Yes.
The timing. Yes.
Yeah.
So if you have two markers for coffee, let's say, one that is very acute and recent, and then one that is a longer time.
Oh, I see. Yeah. If you have two markers of the
when you
same
sample,
food,
do you see them both or only one?
I see. Yeah, that's an interesting idea. Thank you.
Most welcome.
Great. Thanks, John. So moving forward, is a database for reference and index for metabolite markers specifically to food?
Everything. I mean, there are more and more well-thorough databases now, right? So it's also the FoodDB, but I think HANDB it's still the best. FoodDB. Frankly, I didn't do identification in the past 2-3 years. I also come out of 2 maternity leaves, so maybe I say also something not fully accurate, because I think the field and the databases are actually developing at a high rate. But essentially, it's still everything.
Yeah. Okay. Moving on, I have my own question here. How different are biomarkers in different food cultivars? So you had shown a bunch of reviews that the food ball had, produced, and some were like on berries and some were on some kind of, different specific foods. But do you see like differences of the same food in different group, or like different food cultivars in the same food groups, like Granny Smith apple versus like a Red Delicious apple? Or is it just generally like just an apple, Do you
Yeah.
see that difference?
I think so far we are, I'm not over all the different fruits and vegetables, and I think it's there where this matters the most. So I was not the one working on those reviews. But I know it matters. I know that I can imagine that PTFI is going to be actually pretty good at signaling those out, the differences between the different varieties. Yeah, but they do, and essentially it's we have some chicken markers that are only found in Ireland, and we don't see them in Denmark, and that's mainly because of the different animal feeds, right? So I imagine that that is the case for more foods.
Yeah, yeah, that makes sense. There's several questions about people and food because, we're the ones that are eating it. So how does BFI differ between populations? For example, healthy versus acutely and chronically ill individuals, younger versus older adults, just the different type of people who are eating foods.
So we do see, we don't know for most. It's also because, all the development, I mean, it usually happens in healthy individuals, most of the things we do. But there are differences between men and women in some enzyme-based, and there are also in people with pre-diabetes compared to healthy. So we could actually see some of these differences when we try to validate some markers in this previous study, which was actually a pre-diabetes population. And then we had a lower index of lower levels of some of the markers. Carnosine was one of them.
Yeah, that makes sense. Okay. Is there any variation of metabolites among target groups, ages, and sex?
Yes. Those are the main, actually, some of the mains, along with microbiota and along with, but ADME is influenced by gender and age as well. So yes.
Okay. And then comparatively, BFI requires voluntary participants and ethical clearance to anthropometry methods, in which case is more effective?
This question, and repeat it, please.
So it says comparatively, BFI requires voluntary participants and ethical clearance to the anthropometry methods. In which case is this more effective?
Anthropometric measurements. So this means body composition.
Methods. I believe so, yes.
I mean, anthropometry is body composition. How is this related to BFI?
Yeah, I believe that's what the question is asking. If not, we can move on to the next one. So there were a couple questions going back to sample type. Do the sample type we take, urine, blood, depend on the type of biomarkers we intend to assay? It's a good question.
I mean, I wouldn't say that if we have biomarkers, certain biomarkers that are discovered in urine, we don't expect to see those in blood. I think actually the two biological samples are they are overlapping, but they're also complementary. So I would look for what they are. Of course, we only have the science for what has been done. And sometimes you see them in both and sometimes you don't.
Great. And then the last question here in the chat, it says, what about inflammation impacts on the quality and quantities of metabolites assessment using biological samples? So how does inflammation in the body, affect the biomarkers that are resulting from the food that we eat? It's a big question.
Big question. Food affects inflammation. Inflammation, you would say it's a bit the other way around. I'm not sure.
Yeah, it's a big question. Some foods can cause inflammation as well, in certain individuals. So, it's kind of like cause and effect type of question. Well, that is the last question. If there are any other questions feel free to put them in the chat.
Anyone else who wants to speak up? Arpana, you get quieter and quieter your voice gets, or your sound, it's hard to hear.
Oh,
I get closer and closer to the speaker.
sorry about that.
So anyone else who wants to raise a question, congratulate Catalina to her professorship and to an excellent talk. Anyone else who would like to say something?
Thank you.
All right, well then, thank you so much for staying, late in the evening in Europe with us. Thank you, that was excellent. We hope to see you soon in one or the other meetings.
Hopefully, yes.
And wish you very well and look forward to your next papers.
Yeah.
Yes. Look forward to writing some more. Thank you so much. It's been it's been a really nice opportunity, and I'm happy I was able to actually be with you guys today. And have a nice day. I'm going to sleep, but we do enjoy summer while it lasts, I hope.
Yeah.
There's still some summer in California.
Great. Thank you, everyone.
Bye. Thank you.
Bye.